Welcome to this video. In this video we're going to talk about haemostasis. Okay, so this video will be split into several parts, and all of the parts will be found in order in the playlist on the cardiovascular system. Okay, so this is the first part of the video, but if you're looking for all of the following parts, that's where you can find them in the playlist on the cardiovascular system. Okay, so what are we going to do in this first part of this video then? We're going to start off by looking at the definition of haemostasis. We're going to understand what are we actually going to be studying, and we're going to have a look at the broad overview of what haemostasis is actually going to involve. Then we will begin our in-detailed study of the process, and we'll begin with the first part of it, the logical first part to begin with, which is platelet adhesion. Okay, so let's start off with the definition of haemostasis. So the definition of haemostasis is ending hemorrhage. Okay, stopping hemorrhage. And of course hemorrhage means blood leaving the circulatory system and going out of the circulatory system, either into the tissue fluid or potentially out into the external world. If you've cut yourself open, then your blood can leak out into the external world. Okay, so this is the situation then that we're going to be dealing with. We're going to have a blood vessel here, and this is going to be, a medium-sized blood vessel. Hopefully it won't be an enormous one, more a sort of medium-sized one that, if you get injured and get a bruise, you might have broken open. Okay, so a medium-sized blood vessel, but what I want to stress is we're not really talking about tiny little blood vessels like capillaries here. We're talking about medium-sized blood vessels. Okay, so here then, let's say this is your medium-sized blood vessel here, and what's now happened to it is some sort of injury has occurred and you've got a hole in the side of your blood vessel here. Okay, now of course what can happen is the blood from within the blood vessel can then leak out, and either it will go into the surrounding tissue fluid, or if you've actually got an open wound, then it might leak out into the external world, and this is of course hemorrhage. Okay, so hemostasis is the physiological response that is going to stop this, because you do not want your blood leaving your circulatory system. The blood is essential. It's the medium through which we deliver oxygen and nutrients all around the body. It's the transport medium. Okay, if you lose it, then you lose the ability to transport oxygen and nutrients around the body. Okay, so you want to prevent hemorrhage, and the body has a physiological response to deal with an injured blood vessel, such as the one that we've got here. Okay, so what I now want to do is give you the broad overview of what is going to actually happen to prevent blood leaving the circulatory system through this hole that we've got in this injured blood vessel. Okay, so you're probably aware of what's going to happen. One of the key things that's going to happen is that you are going to form a plug in this hole. Okay, the blood is actually going to form a solid mass that will plug in this hole, and the process when the blood turns from being a liquid to being a solid has a fancy name, and that is what people mean when they say coagulation. So coagulation is the process where the blood turns from being a liquid to being a solid, and then can form, of course, what we would call a blood clot, okay, which is a solid plug made from the contents of the blood that will fill in this hole and prevent further blood escaping through that hole. Okay, as we'll see later, there is a better name, blood clot. Professors will advise that you steer clear of the word blood clot, because when you say blood clot, it isn't clear whether you're talking about what we're talking about here, which is a physiological blood clot, a good blood clot, where you've got the blood forming a clot in a hole in the side of a blood vessel, or whether you're talking about a pathological blood clot, which is where a blood clot forms in a perfectly fine vessel, a vessel that doesn't have a hole in it, and that's what's known as a thrombus. Okay, so we will actually refer to this sort of blood clot, a good blood clot that's actually filling in a hole in the side of a blood vessel, by the fancier terminology, a hemostatic plug. Okay, so people prefer you to use hemostatic plug when you mean a good blood clot, a physiological blood clot that is actually plugging in a hole in the side of an injured blood vessel. Okay, right, so that's one of the key things that is going to happen. The blood, the constituents of the blood are actually going to change from being a liquid to being a solid. That's known as coagulation, and we will form a solid hemostatic plug in this hole in the side of our blood vessel. Okay, and of course we will be going through in huge detail the process by which the blood actually coagulates and turns from being a liquid to being a solid that can plug in the hole in the side of this blood vessel. Okay, but there's another really important aspect of hemostasis that often people forget. This is the iconic one, the one that people have no trouble remembering. Okay, but the other very important part of hemostasis is that this blood vessel that has the hole in the side of it, it will also undergo vasoconstriction. Okay, now of course vasoconstriction is a long word to mean the blood vessel will constrict down and reduce the diameter of its lumen, so it will become much narrower. Okay, and of course if the blood vessel becomes much narrower, then there will be much less blood flowing through that blood vessel, and if there's less blood flowing through that blood vessel, then even if you've got a hole in the side of it, then your hemorrhage is hugely going to be reduced. So vasoconstriction is an important part of hemostasis. It's an important part of the physiological response to having a damaged blood vessel in this way, and it shouldn't be forgotten. It's very important for stopping hemorrhage. Okay, so coagulation and vasoconstriction, and what we are now going to see is exactly how this is brought about. So how does the blood recognize that there is a hole in the side of the blood vessel? Okay, what actually triggers the blood to undergo the coagulation process? What actually triggers the blood vessel to vasoconstrict? How is this all coordinated? What are the mechanisms? That's what we're going to begin with now. Okay, and we're going to go for an in-depth discussion of the mechanisms, but that's the broad overview of how you actually prevent hemorrhage. Okay, so beginning now with the actual in-depth mechanism, the first thing that I want to begin with is to introduce an absolutely key player in all of this, which are platelets. Okay, so we're going to spend a little bit of time just introducing platelets because they're absolutely essential in this entire process. Okay, so platelets is the most normal name that you'll hear people call these things. However, there is a fancier name. People sometimes will describe them as thrombocytes. However, despite this name, despite having sites here, okay, they are not actually considered cells. Okay, they are actually fragments of cells. So let me explain this. So firstly, what I want to do is I want to draw a platelet alongside a red blood cell. And I suppose the first thing I should say, I'm just assuming you know this, but platelets are a constituent of the blood. So in your blood you will have these little structures known as platelets, and they are like tiny cells, although they are not considered cells. Okay, so let me show this. So first I'll draw a red blood cell. So I'll draw my red blood cell here. So here's the biconcave disc of my red blood cell. So this is my RBC or erythrocyte, whatever you want to call it, and I'll just colour it in red to make it look genuine and easily recognisable. Okay, so there's my red blood cell. So now what I want to do is draw a platelet so that you've got a comparison for size. So a platelet would be around this sort of size, so much smaller than a red blood cell. Okay, so here is my platelet or thrombocyte, whichever you would like to call it. And it looks like a little cell. It's got a cell membrane. Okay, so this line that I've drawn here, this is the cell membrane. It's got some cytoplasm inside there, but of course it has no nucleus. Okay, and in fact, really the way you should think of it is as a little fragment of a cell, and indeed it actually is a little fragment of a cell. It's made by these enormous cells that are in the bone marrow called megakaryocytes, and they throw off little portions of their cytoplasm encapsulated by portions of their cell membrane. So let me just draw one of these. So let's have one here. So let's say this is a megakaryocyte, and let's say up there it's producing a new platelet. So in the bone marrow then, which is the main place where blood cells are being produced all the time, one of the special types of cell that you have in the bone marrow is known as a megakaryocyte, and these are very large cells. And what these do is they are all the time producing platelets, and what they do is they pinch off a little bit of their cell membrane, as I've sort of tried to show here. Okay, and they gradually pinch it off, and it will contain a bit of cytoplasm inside there, and then it will pinch off like this to form a little platelet. So platelets are actually produced by pinching off little bits of cell membrane along with little bits of cytoplasm from these massive great cells inside of the bone marrow called megakaryocytes. Okay, and you'll have lots of these platelets. They are a common structure found within the blood. So, as I say, they're not considered a cell, they're considered a fragment of a cell. So platelets, often you'll hear them described as fragments of cells. Fragments, literally, of megakaryocytes here. Okay, now platelets play an extremely important role in the coagulation process, in the process whereby the blood will recognise that there is a hole in the side of the blood vessel and then turn from being a liquid to being a solid. And the first part of that is a process known as platelet adhesion. Okay, so what we need to understand is how can platelets tell the difference between when they're going through a perfectly intact, beautiful blood vessel and when they've suddenly got a hole in the side of the blood vessel and they are travelling through a hole in the side of the blood vessel. Well, what you have to understand is that platelets usually have a very boring life. All they see is the other constituents of the blood and the endothelial cells of the blood vessel. So remember, if I just draw now a cross-section of my blood vessel here, okay, so let's say this is my blood vessel. Remember, we are considering a medium-sized blood vessel. We are not just considering a capillary. Okay, so it will have lots of layers in its structure. Okay, and the innermost layer will be the endothelial layer, the layer of endothelial cells, which look like this, and those are the cells which the platelets are actually going to come in contact with. So the platelets will usually be in the lumen of the blood vessel here. So all they will be seeing are the endothelial cells and whatever the endothelial cells have on their surface. So they don't actually usually get to see that many molecules. Now, this is the idea. When you suddenly get a hole in the side of your blood vessel, so we've now cut out this portion here and we've got a great big hole in the side, what will now happen is these platelets will move through the hole and they will rustle. They might, bang up against the boundaries of the hole. So imagine you've tunneled down through the wall of the blood vessel. You've got now a little tunnel in there, and now of course there's the walls of the tunnel are now these exposed portions of the walls of the blood vessel here that weren't previously exposed, and now the platelet's going to be banging against those portions. Now those portions have a very different molecular profile to the endothelial cells. They will have molecules in their extracellular matrices, for instance, which these platelets were never allowed to see before. Previously, the platelets had a very censored life. What they were allowed to see was what the endothelial cells showed them. Okay, they weren't allowed to see a whole bunch of molecules, and now that they're suddenly banging up against all of this tissue here, they're going to see a bunch of molecules that they never saw before, and that is what lets the platelets know that something is wrong and they need to change their behaviour to cope with this. Okay, so that's the fundamental answer as to how the blood is going to tell the difference between an intact blood vessel and a hole in the side of the blood vessel. The answer is that there are loads of molecules that the blood would not usually see when it's inside a blood vessel that it is now going to see as it travels through this hole inside the blood vessel. Again, that's what's going to trigger the coagulation process. So let's actually have a few specifics here. So platelets on their surface have receptors, okay, which are called platelet receptors. And these platelet receptors mainly belong to the family of receptors known as integrins. So I'll just give you a little bit of a talk about integrins. So integrins are a family of cell adhesion molecules, okay, and they are made up of two proteins, two different types of protein, an alpha chain and a beta chain. So after I've just underlined these words, I will draw a picture of an integrin. Okay, so let's say this is the cell membrane here, so this is the phospholipid bilayer of the cell membrane. Integrins are going to be made up of two separate proteins, dimerized together in this way. Okay, and just to keep it nice and simple, I've just drawn two rectangles to represent these two proteins. So one will be called the alpha chain of the integrin. Okay, so let's say this one in green here, this is the alpha chain of our integrin. And the other will then be called the second letter of the Greek alphabet, beta. So here in orange, this one is going to be the beta chain. Okay, so they form these heterodimers, hetero meaning different, dimer meaning two-membered structure. So integrins are heterodimers, and platelets have a lot of different integrins on their surface, and these integrins can bind to ligands, so ligand molecules can bind to the extracellular aspect of the integrin, and then it can have effects intracellularly in the normal sort of dogma of the way receptors affect cells' behaviour in response to their ligand. Okay, so platelet receptors are going to consist of these integrins which will have different ligands, and some of these platelet receptors are going to bind to molecules that are now going to be exposed when we've got this hole in the side of our blood vessel. So let me give you some examples here. So, one of the platelet receptors that's very important for you to know about is glycoprotein. The GP is for glycoprotein 1A/2A. So what does the name mean here? It's referring to the fact that the integrin is a dimer. So one of these will be the glycoprotein 1A and one of them will be the glycoprotein 2A. I'm afraid I don't know, and neither do I think it's particularly important which of these is the alpha and which is the beta in this case. But one of these will be the alpha chain of the integrin and the other will be the beta chain of the integrin. Okay, so this is our name for the overall integrin. So this is one of these platelet receptors that is on the surface of platelets, and the molecule that this has as its ligand is collagen molecules. It can bind to collagen molecules. Now integrins are often used as cell adhesion molecules, so they can often bind to collagen molecules because they're often the means by which cells attach their cytoskeletal components to the extracellular matrix. Okay, so here the platelets are going to have this glycoprotein 1A, 2A on their surface, and this is going to be able to bind to collagen molecules. Now, when they were inside the blood vessel, they did not ever get to see collagen molecules. The endothelial cells never let them see collagen molecules. Now that they're going through this cut open, drilled in hole in the blood vessel wall, they're going to come into contact with loads of collagen because collagen's everywhere. It makes up the extracellular matrix. So in between cells in your body you have an extracellular matrix which basically looks like a spider's web between the cells that is holding all of the cells together, and one of the major components of this is collagen. Okay, so now that we've got this drilled open hole here, these platelets are going to easily come into contact with collagen, whereas the endothelial cells previously were shielding them from collagen so that they never saw that. Okay, so now of course what's going to happen is these glycoprotein 1A, 2A integrin molecules on the surface of the platelets are going to bind to this collagen, and that's going to end up binding the platelets to the surface of our hole in the side of the blood vessel. Another example that I believe it worth you knowing is glycoprotein 1C/2A. Now this also binds to other components of the extracellular matrix. This one binds to components of the extracellular matrix that are slightly less famous, but still noteworthy, fibronectin and laminins. Okay, so fibronectin and laminins, these are proteins that are also key components of the extracellular matrix, key components of this spider's web of proteins and carbohydrate molecules that hold cells, neighbouring cells together, okay, and are very important, therefore, in the structure of the body. Okay, and again, platelets usually are never allowed to see these molecules. The endothelial cells shield them from these molecules. So now that the platelets are banging up against the surface of the hole in the side of the blood vessel, they are going to see these molecules because these molecules are everywhere, and this platelet receptor, the glycoprotein 1C2A, and I feel like I should just write out glycoprotein in case anyone doesn't know how to spell that. So GP here is standing for glycoprotein. So what will now happen is these glycoprotein integrins will bind to these molecules of the extracellular matrix, and what's now going to happen? You're going to end up with platelets binding all over the surface of this hole. So let me draw it from another aspect. So let's say this is our blood vessel from the side here. Here is our hole. Okay, the disadvantage to this is you can't see the depth of the hole, but I hope this will get the point across, and what you're going to end up with is platelets lining the surface of your hole, basically. Okay, so all of this exposed surface here, where you're going to have collagen and fibronectin molecules now exposed to the platelets that are coming through this hole from the lumen into the extracellular space, you're going to end up with platelets lining the surface as their platelet receptors bind to these molecules that they were previously never allowed to see, which are their ligands. And this process where the platelets bind to the exposed surface of the blood vessel wall, this is known as platelet adhesion. Okay, and this is one of the first parts of beginning the coagulation process, that platelets are going to adhere to the wounded, exposed blood vessel wall structure here. Okay, right, so that's the first part, platelet adhesion. What we will do in the next video is we'll move on to the second part, which is going to be called platelet activation. So what's now going to happen is these platelets are going to change their behaviour hugely. Okay, they're going to trigger loads of other platelets that haven't actually even come into contact with the collagen or the fibronectin and laminin molecules to also undergo the same behavioural change. And then these platelets that have been activated are all going to aggregate, because as you'll see, what do we want to form here? We want to form a plug. At the moment we formed the outer lining. We need to somehow fill in the bit here, and we're going to do that. We're going to form what's called the platelet plug, which will form part of the overall hemostatic plug. It's not very strong on its own. It will need other components to actually make the full-on hemostatic plug. Okay, but we are going to completely plug this hole with platelets, so we're going to somehow have to get the platelets to stick to each other, and indeed that's what platelet activation is going to do. We're going to need a chain reaction now to get loads of platelets that haven't adhered to the surface of the hole here to actually start binding to other platelets, so that we can form a nice big plug of platelets in the hole here. Okay, so we'll have a break and do that in the next video.